We are starting chapter 14 today. Chapter 14 is all about human heredity. So, we're going to look at chromosomes and we're going to look at some genetic disorders. You're actually going to do a research project with this chapter where you get to choose a human genetic disorder and research it, and then you'll tell the class about it. And then we'll also look at the Human Genome Project at the end of this chapter, which was um, a project that involved lots of scientists from around the world trying to sequence the entirety of the human DNA and figure out where we have particular genes located. And so we actually know where genes are located on like every chromosome. We know a lot of them. Anyway, we probably don't know all of them. Um, and there's still a lot to learn there, but we'll go through kind of how we learned a lot about the human genome, which is like the entirety of human DNA. Um, so you should have gotten your calendar and your review for chapter 14. Um, you might need to make some adjustments to that calendar. You guys might have needed to take some extra time with something or whatever. So if you need to make adjustments to the dates, keep that in mind or change those things. But let's get started with chapter 14 and human heredity. So we are looking today at chromosomes and human chromosomes specifically. And so we've talked about chromosomes for a while now. We started talking about them with mitosis back in chapter 10. We talked more about chromosomes in chapter 11 when we talked about meiosis and genetics and all of that. So this is not something that's new, but we're going to talk about it in a little bit more in depth. So we're going to start by looking at karyotypes. So I was just talking about the Human Genome Project. So your genome or genome, depending on where you're at, is all of the genetic information that an organism carries in its DNA. Okay, so everything, all of your chromosomes, all of your DNA. We can take a picture, basically, of your DNA, of all of your chromosomes. And it's called a karyotype. So this is a human karyotype. And the way that they do this is they take cells that are in mitosis, okay, so that the chromosomes are condensed, and they find all of the pairs of chromosomes. Okay, so this is diploid, right? We have two sets, and this happens, they take these cells from prophase. So you have two sets of all of your chromosomes, and you can see they make that kind of X shape that we always draw for our paired chromosomes. So we have 23 pairs of chromosomes. So we put them in order, we pair them all up. They get stained, which is where this coloring comes from. So based on their staining pattern, we pair them up. And then also based on their length and their where the centromere is. So there's several features that we can look for to identify which chromosomes should match. And then we put them in order based on their length. So we start with the longest ones and go to the shortest. So you can see some of them have very different shapes. Some of them, like 22 down here, has the centromere is really close to the top, whereas number 1 is like right in the middle. And so that centromere placement is very different on each of these chromosomes. So we line them up from the longest to the shortest, and then we put the X and the Y. So this is a human male because we have a Y chromosome. You can see the Y is much smaller than the X. And we'll talk about that a little bit here in just a second. And you're going to do a lab with karyotypes. So the X and the Y are the chromosomes that determine gender. So we call them the sex chromosomes. They determine your sex, your gender. If you get 2 copies of an X chromosome, then you're female. If you get an X and a Y, you're male. And so this Punnett square showing why approximately 50% of offspring are female and approximately 50% are male. So mom has two X chromosomes, so they go through meiosis and you produce egg cells that each have one X chromosome. And then dad has an X and a Y, so his cells would go through meiosis and produce sperm cells that have either an X or a Y. So it is actually dad that determines gender, whether or not dad passes on an X or Y, because mom can only pass on X. So this is comparing the X chromosome to the Y chromosome. Your X chromosome carries lots and lots of genes, carries at least 1,200 genes. And so the X chromosome, while it does have genes that are important for the development of female characteristics, it also has genes for other things as well. So there's a few of them that are labeled here. There's a gene for muscular dystrophy, which is a genetic disorder, and it's one that you can choose to research. Hemophilia, colorblindness, those genes are found on the X chromosome. And then your Y chromosome only has about 140 genes on it. Most of them are associated with male characteristics. Okay, so any traits that are on the Y chromosome get inherited directly from father to son. The X chromosome, though, you could get from, if you're male, you got it from your mom. If you're female, then you get one from mom and one from dad. All of the other chromosomes are called autosomes or autosomal chromosomes. So you have your two sex chromosomes, the X and the Y, and then everything else is called an autosome, and they just get a number. Okay, and remember, we number them when you put them in order from the tallest or the longest chromosomes. Those are number 1 to, in humans, you would go to 22. Okay, so this is 44 because you have pairs of each you have 2 of each of those numbered chromosomes. So we've talked about human traits a lot, especially if you think back to chapter 11. So we're going to kind of bring out some of that again. So in humans, we can have traits that are passed that are just that simple dominance, right? You have a dominant or a recessive. So for instance, the Rh blood factor, whether you have a positive or a negative blood type, positive is dominant to negative. So if one of your parents has a positive, or you get a positive Rh factor from one parent, then you are positive blood type. We can have codominance and multiple alleles, and we've talked about this example a little bit. This is your blood groups. Okay, so whether you are type A, B, AB, or O. So A and B are dominant to type O, but they're codominant to each other. So if you are type A blood, you either got 2 copies of the A allele, or you got an A and an O. If you're type B blood, you either got 2 copies of type B, or you got a B and an O. And so if you're type A, you are going to attack any type B blood. Okay, so it's safe to get type A blood, but you would attack anything where you see the type B. Okay, so because what this is coding for is a protein on the surface of your blood cells. And so that protein helps your body to recognize whether something is part of yourself or not part of yourself. And your body really doesn't like things that are not part of itself, and it attacks them. So if you have type A blood, then you're going to attack type B because it's not part of you. And so then you can get really sick. This is why transfusions, they try to match your blood type as closely as possible. If you have a type A and a B allele, that you get A from mom, B from dad, or whatever, then they're codominant and you show both. And so then you don't actually attack either one because your body recognizes both the A and the B. So you could take type A blood or B blood or type AB. AB is actually the rarest blood type. Type O The only way to be type O is to get 2 copies of the type O allele. And really, the type O allele is that you don't have any proteins. You don't have either the A or the B. You have no proteins on the surface of your body, so you attack A and B. But type O blood is actually the most common blood type, at least in the US. It changes a little bit if you go like worldwide or go to certain subcultures or populations around the world. Um, those frequencies change a little bit, but for the US, O is the most common and AB is the least common. Okay? Just to kind of show you that, we've talked a lot about recessive does not mean rare. So we have, this is the recessive condition, having type O blood, but it's the most common as well. We can also have traits that are linked to the sex chromosomes. And it's called sex-linked inheritance, or a lot of times we talk about X-linked traits. So any genes that are located on the sex chromosomes, the X or the Y, are considered to be sex-linked. If you have a gene on the Y chromosome, it's only going to be found in males because they're the only ones that get a Y chromosome, and it gets passed directly from father to son because that's where you get your Y chromosome. Any genes that are found on the X chromosome are found somewhat equally in both genders, but it depends. So if there's a recessive X-linked condition, it's actually more commonly found in males than in females. And that is because males only get one copy of the X chromosome. So if you get an X chromosome with an X-linked recessive condition like colorblindness, red-green colorblindness is X-linked. So if you get one copy of the recessive X-linked colorblindness trait, you're going to express it because you don't have anything else to mask it. If you're female and you get one copy of that X-linked trait, well, you have another X chromosome. So you could get the dominant form, which in the case of colorblindness would be normal vision. So you would have to get 2 copies of that recessive trait in order to show it if you're female. Okay, because you have another X chromosome that could cover up, could have the dominant version and cover up the X-linked trait, okay, or cover up like colorblindness. So it is more common to find red-green colorblindness in males than in females. It's not impossible for a female to have it. In order for a female to have red-green colorblindness or any X-linked recessive condition, dad has to have it and mom at least has to carry it. Okay. And then then your daughters would also at least have a chance of having that trait. Okay. And so if I need to, I can explain that a little bit better, but you do have access to some other videos that'll show that. But just knowing that those recessive X-linked traits are more common in males because they don't have a second X chromosome to cover up any recessive conditions on one X chromosome. Okay, so females have that second chance of having the normal condition or having the dominant condition. Another thing that happens, so females have 2 X chromosomes, but for a lot of our genes, having 2 X chromosomes is actually too much. So what happens in females, okay, it doesn't happen in male cells because they only have the 1 X chromosome. But in females, in most of our cells, one of our X chromosomes will randomly turn off. Okay. And what happens is that randomly in every cell, one of your X chromosomes condenses. It forms this dense region. It's called a bar body. I think named for the person who discovered that this is what's happening in your cells. So you're kind of randomly in all of your cells expressing either one X chromosome or the other. That's still enough. So like if you are a carrier for, let's say, red-green colorblindness, it's still enough that one of your cells, or that one chromosome, or enough of your cells should still um, have, if you're just a carrier, right? So you have normal vision on one X chromosome and color blindness on the other, even if randomly throughout your body, um, you know, approximately half of your cells would have the normal vision and approximately half would be showing color blindness, that's usually still enough that your vision is normal. But one way we do see this is in cats, specifically calico cats. I closed the door so you guys don't get to see mine. She's not going to be here and all up in my face. So calico cats are almost always female. And so the calico cats are the ones that have the orange and the black, and then they're usually like their belly and their feet is white. Okay, so having this tricolor, these three colors that are showing up, they're almost always female. And that's because the gene for their fur color, for the orange and the black, is on the X chromosome. So if they get two copies of the black fur color, they would be black. If they get two copies of the orange, then you get the orange cat. If they get a black and an orange, then in all of their cells, randomly, one of those is turning off. So some of their cells start to express orange colored fur, and some of them start to express black colored fur. So that's how you get calico cats, and none of them really look the same. They all have all of these differences in their fur coloring because it's random. Okay. All right. I believe this is the last thing we're doing is pedigrees. So a pedigree, and you may have seen this before, it's like a family tree. And it's one way that we can track traits. So you can track traits in any organism using a pedigree. So a lot of times it gets talked about like with animals, especially if you want be able to prove that you have a purebred, whatever kind of dog or horse or whatever. But we can use it to track traits in humans. And so based on the pedigree, we can determine if the trait is dominant or recessive, if it's on an autosome or a sex chromosome. And so the way that these work is that the squares are for males and the circles are females. A line across is indicating like a marriage. And then you would draw a line down, and then you would have kind of a bracket shape to their kids. So these people have 3 kids. They have a son and 2 daughters. And then their son got married and one of their daughters got married. And so then we had 2 kids here and 3 here. And so then if it's shaded, then they show the trait. So this particular pedigree is tracing the presence of the white forelock. So that's people who like right here have white hair, just like right at the very front of their hair is white. Okay, so we have 3 generations here, right? So granddad has this trait, but grandma doesn't, right? And so then both of these individuals have the trait, so they managed to pass it on to one of their offspring, but not both. So if you were to be examining this, would you say that this trait is probably dominant or probably recessive? So think about that for a second. So if it was dominant, right, then grandma here would be, let's see, what letters shall we use? We'll use A. Okay, so if it's dominant, right? Let me change color because that you can't see. Nope, that's not really good. Let's go with that. All right. So if granddad has it, he at the very least is a capital, right? If it's dominant. Okay, and then grandma here would be little a, little a. Okay, so both of these would have to have a capital A, right? And we don't know enough information to be able to predict what granddad is here. Um, if this is a dominant trait, right? And so eventually, and this is a little bit beyond what you guys are expected to do, but you could be eventually asked in a later biology class to be able to predict like what type of inheritance it is, or even if it's linked to and linked to a sex chromosome, linked to the X. Um, so it's definitely not linked to Y because there are females that have it. Uh, so it's kind of like a little logic puzzle. So you're like, okay, well, they managed to have a daughter that doesn't have it, right? So if this is the dominant condition, then dad must be big A, little a, right? Or grandpa must be because she doesn't have it. Okay, so it's a little bit of a logic puzzle to figure out what everybody is. And sometimes you get through it and you realize, oh, it's probably not the dominant condition of it. It was actually recessive. Uh, Sometimes that makes more sense. It doesn't make sense for this one. It should be dominant um, because if these two individuals were, let's say it is the recessive condition, right? If these two, then they would have to be little a, if it were recessive. Well, but then how would they have a child that would have a big A? Right? How would they have a child that has the dominant form? Okay, so um, most likely this is the dominant. Having that white forelock is actually dominant. And because if both of these individuals are heterozygous, right, they could pass on. It's hard to predict what this daughter would be. She would be a question mark or a blank because we don't know whether she got the big A from mom and a little a for dad or big A from both. We don't have enough information, right? But we know this daughter got the recessive condition of having no, having a normal forelock because she doesn't have the white. But that's how you would fill in a pedigree or use it's a logic puzzle. You just figure out, okay, we start at the top and we figure out what people probably carry. So because um, they managed to have a child without the trait and who would have the recessive condition, they each have to carry an allele for the recessive. Okay, so that's how you do human pedigrees. That's how you work those out. All right, and that I think is the end of. Oh, I did that wrong. That should be the end. Yeah, that's the end of the slideshow. If you guys have any questions, let me know. If you're struggling with this, if I didn't cover something really well, or I was just very confusing, it's possible. But that is the first lesson of chapter 14. So that's human chromosomes. We'll talk about genetic disorders in the next lesson, and then the human genome as a whole in the project and how we've learned more and more about it in the third lesson.